Continuing on with the stomach, we're going to look into the digestive processes that occur in the stomach, and a little more the physiology of how the stomach digestion is controlled. But first off, physical digestion. Sure, the stomach is churning, it's mixing. That's physical digestion. Denaturation of proteins. We mentioned that the hydrochloric acid that makes it very acidic helps to denature the proteins. Denaturing proteins is kind of unfolding them, making them not functional anymore. Enzymatic digestion. Well, your cells are producing pepsinogen, but then the hydrochloric acid helps convert that to the active form pepsin. The enzyme pepsin helps to enzymatically break down those proteins. Now I have up here also that renin helps in infants. That's just something that helps out as they're a little bit younger there. Secreting intrinsic factor. Mentioned intrinsic factor is helping with absorption of vitamin B12. So you have intrinsic factor also coming from here. And last, it delivers the chyme to the small intestines. That's its last goal, its last task. Move it along to the next phase of the digestive process. So your stomach really does quite a bit. So we'll look into more of how this all works. Neural and hormonal mechanisms. Now we mentioned, as far as the hormonal in the last video, we mentioned two different hormones coming from those endocrine cells. But now we also have to think about the neural control. That's both that gut brain, that intrinsic neural, as well as the central nervous system. But all this is either for stimulation or inhibition of the processes. There's three main steps. First off is going to be the cephalic phase. Cephalic phase is your thinking about food. You see the food, you're smelling the food. It's all before you eat. It's your system getting ready to accept the food. You then have the gastric phase. Gastric phase, 3 to 4 hours long, give or take. It varies, obviously. But now this is when the food enters and you're talking about more in the stomach. And then you have the intestinal phase. In the intestinal phase, that's when food starts to get into the duodenum, so small intestines, and starts to work its way through there. So we're looking at 3 different phases here of your digestive process through your stomach and then intestines. Now this one has a lot going on. You can see it's from the cephalic phase, brain, you have the gastric phase, stomach, you have the intestinal phase, intestines. So I'm going to blow it up full screen to make it easier to see. We need to go through some stimulation and inhibition events. For stimulation or stimulatory events, we mentioned already, sight of food, thought of food, smell of food. It's stimulating the brain, that cerebral cortex, sending information down into your stomach, down that vagus nerve. But the inhibitory aspect, you can lose your appetite for many different reasons. One potential loss of appetite is if you have a state of depression going on. That now again is the cerebral cortex, but in this case, you're now inhibiting basically what's going on by not sending stimulus down to your stomach. So by not sending the stimulus down, not sending the signal down that vagus nerve, you're causing a lack of stimulation, in essence, inhibition. You then have the gastric phase. The gastric phase could be looking at the stomach distension. Distension is stretching. So when you start having the stomach stretch out, that's a local reflex saying, hey, the stomach's stretching, there's food in here, we need to do something. So again, it can go from local reflex, but also send it up to the brain. Medulla figures this out, hey, we need to act back down the vagus nerve. Or looking at the G cells, or looking at the chemical aspect here. So peptides and rising pH, so it's not as acidic, telling your stomach to, hey, we need to act, there are things coming in, and this is now changing the environment. All of this is stimulatory. On the inhibitory side, excessive acidity. So if your stomach is below a pH of 2, maybe it's down to 1, where it can happen sometimes. Your stomach just doesn't feel right because it's too acidic. So the secretions of gastrin are declining. Or it could be also, again, go back to emotions, the brain. Just like we talked about depression in the first phase, the second phase could be emotional upset. Sometimes when you're very upset over something, it's a very traumatic event, you don't feel like eating, your stomach feels off. So that's the sympathetic nervous system being stimulated. Sympathetic nervous system is overriding the parasympathetic controls. And then the intestinal phase. So if you have low pH in your intestines, because that's not what should be there, that's the stomach. So a low pH in the intestines, specifically the duodenum, means that, hey, we have some secretion that just came from the stomach. That was acidic. It's time to react. So we're now going to have more gastric release. We're going to have more action of the intestines. On the inhibitory side, distension of the duodenum. We're thinking, hold on, that's inhibiting. Yeah, because if it distends too much, you don't want to extend any more food. You don't want more of the chyme coming in if it's already distended. So you're having this inhibitory effect back to the stomach saying, hey, stop, don't send any more food down here. We don't want any more chyme. So if you have more fatty acids, which are slower to digest, partially digested food in the duodenum, it's all about telling the stomach saying, hey, don't send anything else. We're still working here. You have to have the duodenum go back down to a smaller size, have the acidity go closer to neutral, and make sure the chemoreceptors aren't picking up partially digested foods. Once that happens, then you go back to the stimulation to release more chyme. It's all about regulating how much chyme comes out of the stomach. So all these processes, they're all trying to make sure you're controlling the stomach secretions, controlling the release of chyme out of the stomach. And these three chemicals, acetylcholine, histamine, and gastrin, all three are going to be necessary and to cause stimulation of the parietal cells. Now, they're through this basically secondary messenger system, but their goal is to secrete hydrochloric acid. But in order to secrete that hydrochloric acid, all three chemicals have to stimulate parietal cell to say, hey, it's time to secrete. It's a way of trying to make sure your stomach doesn't become too acidic because you don't want to have that pH of 1 if you can help it. Now, antihistamines. You may be thinking antihistamines, that's allergies. True, but antihistamines actually block the hydrogen receptors. When you block those H2 receptors, you're decreasing hydrochloric acid. So it actually can help, as a side effect, help with acidity in the stomach if that's a problem. So here's how we're looking at the secretion and production of hydrochloric acid out of the parietal cells. But again, there's a lot here, so let's blow this one up to really see what's going on. So first off, you can see we have the blood capillary, the chief cell, the big parietal cell right in the middle there, and then we have the stomach itself, the lumen. So that's where things are going, that's the target. So first things first, we need carbon dioxide. Thinking, hold on, we're not talking about breathing here. Bear with me. Carbon dioxide is brought into the parietal cell. It then combines with water, and you produce H2CO3. But that's only because of the carbonic anhydrase. The enzyme helps facilitate that reaction. Now that H2CO3 is going to break apart. You break off a hydrogen, and you end up with HCO3-. That's bicarbonate. That HCO3- is going to hang out in the parietal cell for 1 second. We'll come back to that. But let's follow that hydrogen ion. The hydrogen ion is going to go out of the cell into the lumen of the stomach. At that point, now we have the hydrogen ion in the lumen. It exchanged a potassium, but if you notice, potassium simply comes right back around. So that's not much of a difference there. Now, what we have to do, though, is figure out what's going to happen next. That HCO3-, that bicarbonate, is going to work with this transporter, the HCO3-, Cl-, so bicarbonate, chloride, antiporter, and bring actual chloride into the parietal cell. But as a side note here, pay attention. You now have HCO3-, bicarbonate, in the blood. That is a key for buffering the blood, maintaining the blood pH. That is basically what you do to help with the buffering system. Now keep that in mind because we'll come back to that in the respiratory system as well. But that's how we get some HCO3- bicarbonate into the blood. That chloride then is sent out into the lumen, which means now we have hydrogen and we have chloride, both in the lumen. We've made our hydrochloric acid. So all of that, all those processes, just to produce hydrochloric acid inside the lumen of the stomach. Because you can't produce that inside the parietal cell, it would damage the cell. So it has to be produced in the stomach, which has that mucosal barrier, that bicarbonate-rich mucosal barrier to help buffer and protect. So when the stomach starts filling, and I'm sure you felt this where you're eating and all of a sudden you feel, oh, I feel full. Well, what's going on? There's a reflex-mediated receptor process here. The idea is, as your stomach is filling, it can stretch a little bit, but there are limitations. So it's called what's called gastric accommodation. Gastric accommodation is talking about the plasticity of the smooth muscle. What that means is the smooth muscle, instead of constricting, it can actually stretch out. And as that smooth muscle stretches, it allows the stomach to expand and accept more food. But there is a limitation. It doesn't just stretch forever, but there is that ability to stretch and accept more food if necessary. So now let's look at the gastric contractility. What that's referring to is how does it squeeze? How does it close down? Now remember, peristalsis is going to be when you squeeze on the tube and then keeps that squeezing pushing down. It's trying to propel the food or the chyme in this matter towards one end. Now this happens about 3 times per minute, and that's controlled by pacemaker cells inside the stomach. Those pacemaker cells are setting this basic electrical rhythm. Now this BER, or basic electrical rhythm, is causing that 3 times per minute peristaltic contractions. It's pushing the chyme down towards the pyloric region. Now this is being caused, you're looking at distension and gastric, but it actually can increase the peristaltic actions. So the more gastric, the more distension, the more vigorous these contractions can get, trying to get that chyme out of here and push things through. But they are the most vigorous and the hardest by the pylorus. That's when you're really grinding up that food, that mechanical digestion is kicking in. So once these peristaltic contractions reach the pyloric region, 1 of 2 things happen. Either you deliver about 3 milliliters of this chyme into the duodenum, or it's forced back up. Basically pushes back upwards in the stomach. Now that is not meant to push it up to the esophagus, hopefully, there's that sphincter, but just pushes it back up the stomach, saying, hey, this isn't working, we'll go back up. These controls are all based on duodenum, because if the duodenum already is full and has a little bit of a stretch going on, you can't shove more in there. You gotta wait for it to empty out before the next batch of 3 milliliters roughly is sent through. So what's happening here is the peristaltic wave is pushing down. Then from there, what we're doing is basically grinding, it's mixing, the stomach's contracting and churning that food. Which then, if you have the space in the duodenum, then you have a little bit of this chyme sent through. If not, and the majority of times it's just going to send it back up into the stomach to make sure that you're not overloading the duodenum. And that's all based on chemoreceptors, osmoreceptors, stretch receptors. Those are all figuring out what's going on inside the duodenum and giving feedback into the stomach. Now, how do we regulate this gastric emptying? We've kind of mentioned this already, but just to rehash this to make sure we're on the same page and add a new piece in the mix. We have receptors. Those receptors are chemical receptors and stretch receptors, which we already mentioned. Now, they also are dealing with what's referred to as the enterogastric reflex. What this is going to happen is we're going to have inhibition of gastric secretion based on duodenal filling. we're focusing on CCK. Now, CCK is cholecystokinin. This cholecystokinin, or CCK, is going to decrease gastric motility based on lipid and protein presence. Well, we're saying, why lipid and protein? Lipids and proteins digest slower than carbohydrates. So the assumption is, if they're there, then you're already having a presence of food. But if those are not there, then the carbohydrate should already be long gone. So you're looking at how the actual duodenum is filled. First of all, is it stretched out? But secondly, what's present? So if you have those lipids, you have those proteins, CCK will slow down the gastric motility, preventing more loading into the duodenum. Now, I've already mentioned carbohydrate moves quickly. Fats, proteins move slower. Fat is really the slowest one here. Looking at almost 6 hours, fatty content can sit around. So therefore, a lot of times you have a very fat-filled or lipid-heavy meal, you might feel full a lot longer because it's just hanging around. It's slower to process, slower to leave. So looking at all the different processes in the stomach, a lot about gastric motility and how things are moving out based on receptor feedback.